Let's start by zooming out for a moment. In Module 1, we focused on the immune system structure and functions, its divisions into innate and adaptive immunity, and the major cellular players. Now, we are moving into immunochemistry, essentially the chemistry of the immune system, where we explore the molecules, reactions, and laboratory techniques that drive immunity and allow us to measure, diagnose, and even treat diseases. Immunobiology is the umbrella science that connects cell biology, molecular biology, and chemistry to immune function. At its simplest, immunobiology studies how the immune system's components, cells, antibodies, antigens, cytokines, and complement proteins interact at the molecular level. Think of it as moving from what the soldiers in our immune army look like to what weapons they use, how those weapons are made, and how they are deployed. Section 2, Antigens, Antibodies, and their reactions. If immunology were a play, antigens and antibodies would be the lead actors. Antigens are substances that can be recognized by the immune system. They could be proteins on the surface of bacteria, viral coat proteins, toxins, or even abnormal self-proteins in cancer cells. In some cases, small molecules called haptens can also trigger immune recognition, but only when they attach to larger carrier molecules. More on haptens soon. Antibodies, or immunoglobulins, are Y-shaped proteins produced by B cells, specifically plasma cells, that bind to antigens. The antigen-antibody binding is extremely specific, almost like a lock and key, which is why antibodies are powerful tools for detecting diseases in the lab. When an antigen binds to an antibody, a reaction occurs. These reactions can be visible, such as clumping, agglutination, or invisible but measurable through lab tests. Understanding these reactions is the foundation of diagnostic immunology. Section 3, Concept and Types of Immunity. We have two main forms of immunity. Humoral immunity, mediated by antibodies found in body fluids, humors, produced by B cells. It's excellent at neutralizing toxins, preventing viruses from entering cells, and tagging bacteria for destruction. Cell-mediated immunity is carried out by T cells and other immune cells that directly attack infected cells, cancer cells, or coordinate broader immune responses. Both arms are interconnected. Humoral immunity depends on T helper cells for activation, while cell-mediated immunity sometimes requires antibody support to fully neutralize threats. Section 4, Immunogens, Antigens, and Haptens. Let's be clear with definitions. Immunogen, any substance that can provoke an immune response and also serve as an antigen. All immunogens are antigens, but not all antigens are immunogens. Antigen, any molecule recognized by an antibody or a T-cell receptor, regardless of whether it causes an immune response. Hapten, a small molecule that by itself cannot provoke an immune response but can become immunogenic when attached to a carrier protein. Section 5, Classification and Structural Motifs of Immunoglobulins. Antibodies come in 5 main classes. IgG, the most abundant, provides long-term immunity after infection or vaccination. IgA, found in mucosal areas, like saliva, tears, and breast milk, protects entry points. IgM, the first antibody produced during infection, large, pentameric structure. IgA, involved in allergies and defense against parasites. IgD, found in low amounts, acts mostly as a receptor on immature B cells. Structurally, all immunoglobulins have 2 heavy chains, 2 light chains, a variable region that binds the antigen, a constant region that determines the antibody's class and function. Section 6, Antibody Combining Site and Structure-Function Relationship. The antigen binding site is formed by the variable regions of both heavy and light chains. The amazing part is that antibody diversity is generated by rearranging gene segments during B-cell development, like shuffling words in a sentence to create millions of unique phrases. This genetic shuffling means your body can theoretically produce billions of unique antibodies. Section 7, The Complement System. The complement system is a cascade of proteins that enhance, or complement, the ability of antibodies to clear pathogens. It can punch holes in bacterial cell walls, attract immune cells to infection sites, mark pathogens for destruction, opsonization. Activation occurs via 3 main pathways. Classical pathway, triggered by antigen-antibody complexes. Alternative pathway, triggered directly by pathogen surfaces. Lectin pathway, triggered by specific sugar molecules on pathogens. Section 8, antibody diversity and immunoglobulin genes. Antibody diversity stems from V-D-G recombination, reshuffling of gene segments for heavy and light chains. Somatic hypermutation, small mutations during immune response to fine-tune binding. Class switching, changing from one antibody type, e.g., IgM, to another, e.g., IgG, without altering antigen specificity. Section 9, monoclonal antibodies. Monoclonal antibodies are identical antibodies produced by a single clone of B cells, often fused with tumor cells to make hybridomers for mass production. They have enormous uses. Diagnostic tests, e.g., pregnancy tests, COVID-19 rapid tests. Cancer therapy. Autoimmune disease treatment. Section 10, clinical immunology and immune tolerance. Clinical immunology focuses on diagnosing and treating immune system disorders. A key concept is immune tolerance, the ability of the immune system to recognize and not attack the body's own tissues. When this fails, autoimmune diseases occur. Section 11, antigen-antibody interactions in the lab. Common lab reactions include precipitation, soluble antigen plus antibody visible precipitate. Agglutination, clumping of particles, e.g., blood typing. Hemagglutination, agglutination of red blood cells. Section 12, laboratory detection methods. Immunodiffusion, antigens and antibodies diffuse in gel and form visible lines. Immunoelectrophoresis, separate antigens by electrophoresis, then detect with antibodies. ELISA, enzyme-linked immunosorbent assay, detect antigen or antibody using enzyme-linked antibodies and a color change. Fluorescent antibody technique uses fluorescence microscopy. Radioimmunoassay uses radioactively labeled antibodies. Section 13, hypersensitivity and allergy. 4 main types. Type 1, immediate allergy, anaphylaxis, IgE-mediated. Type 2, cytotoxic, e.g., blood transfusion reaction. Type 3, immune complex mediated, e.g., lupus. Type 4, delayed type hypersensitivity, e.g., tuberculosis skin test. Section 14, major histocompatibility complex, MHC. The MHC is like the body's billboard system for displaying antigens to T cells. MHCI shows antigens from inside the cell to CD8 plus T cells, killer T cells. MHC2 shows antigens from outside the cell to CD4 plus T helper cells. Section 15, cytokines. Cytokines are chemical messengers, the immune system's version of text messages. They stimulate cell growth, direct immune cells to infection sites, control inflammation. Section 16, immunologic disorders. Autoimmunity, the immune system attacks self, e.g., rheumatoid arthritis. Immunodeficiency, the failure of immune function, e.g., HIV, AIDS, SCID. Transplant rejection, immune system attacks transplanted organ. Section 17, immunology of parasitic diseases. Parasites often evade immunity by changing their surface proteins, hiding inside cells, or modulating host immune responses. This is why diseases like malaria remain challenging to eradicate. Immunology and immunochemistry have grown from purely descriptive sciences to powerful tools in modern medicine, from diagnostics to personalized therapies. Understanding these molecular details equips health professionals to interpret lab results, design vaccines, and even use the immune system as a weapon against cancer.